Now, if you recall the title of this session, we're talking about both factorial experimental design and blocking today. Blocking is another aspect that needs to be considered when designing an experiment and conducting the correct analysis. So what is blocking? Blocking is the subsetting of a larger experiment into smaller chunks or blocks, which is often done for practical purposes. For example, if the experiment is too large to perform all at once, or to statistically control for some sort of nuisance variation or noise that can't be controlled for experimentally, such as the position your tissue culture occupies in an incubator, or the cage your experimental rodents happen to share with other rodents, for example. It's common for blocks to be apportioned either over space or time. For example, you might conduct a field experiment over a large plot of land subdivided into smaller units, or you might perform the same experiment over successive days of the week. Blocks are really just a type of factor, but they're one we usually don't care about the effects of scientifically. We do, however, need to account for any noise introduced by blocks statistically so that we can make the correct inference about the factors that we do care about. If we think back to our vaccine trial example, we might rightly decide that having only 12 volunteers wasn't really enough to draw a good conclusion about efficacy of the vaccine. For logistical reasons, the trial might need to run across multiple hospitals, which is a common scenario you're likely to come across in real data. So here we consider 12 volunteers, each randomly assigned to 4 hospitals. In this case, hospital is the block. To account for the variation introduced by conducting the trial at multiple hospitals, we need to include hospital as a main effect in the model. On the left of the screen, here we see the ANOVA table for this kind of design. We have an additional row which accounts for the main effect of hospital. And notice how the sum of squares apportioned to hospital is actually quite large, and actually larger than any variation apportioned to any of the main effects we really care about. If we plot the data, we can see that there's something strange going on with the data from Hospital 4, in that the vaccinated volunteers actually seem to have a worse severity than non-vaccinated ones. And this could indicate, any number of possible mistakes that went on with the trial or the recording of the data that we just can't know about. And had we not known about this, we might have made an inference error, as we'll see. If we compare this to an ANOVA table that does not include hospital as a block, if we just ignore the fact that the patients, the 48 patients came from different hospitals and lumped them all together into one sample, we now see that our inference changes. We no longer find any significant effect of sex or vaccine. And this occurs because that variation that we apportioned to hospital in the blocked model gets lumped in with other residual variation in the non-blocked model. Consequently, our F values decrease and our P values increase. If you'd like to test this for yourself, here's the example code I used to generate these ANOVA tables. You can see exactly how to incorporate hospital as a block by adding it as a main effect in the model. Okay. On this slide, I'm showing you an example of what a block design is not. Had we, for example, taken the same number of volunteers and assigned some hospitals to be the vaccination hospitals and other hospitals to be the control hospitals, this would not be a block design. The reason for this is that it doesn't allow us to make a comparison between the hospitals and, between the vaccine groups and the control groups, because they never occur in the same hospital. And so any variation introduced by being at different hospitals is confounded with the different levels of the vaccine versus control treatment. Next, we'll look at some of the different types of block designs you are likely to come across in the literature. It's completely beyond the scope of this session to cover every possible type of block design. In fact, there's a whole field of statistics that it's dedicated to characterizing all the possible block designs from a theoretical standpoint. But this introduction will hopefully give you a basis for which you can then explore more complicated block designs if they're appropriate in your research. So the first type of block design we're going to consider is a randomized complete block design. As far as simplicity and the ease of analysis goes, this is the ideal block design if it can be achieved with your experimental setup. In a randomized block design, or a randomized complete block design, all factor combinations appear in all blocks the same number of times. Hence, it's a complete design. And this allows us to have equal precision between all possible contrasts to be made. The multi-site COVID-19 vaccination trial we considered is an example of a randomized complete block design if all of the individual volunteers, for example, had been randomized to different hospitals. Another slightly more complicated type of block design is an incomplete block design. In contrast to the randomized complete block design, only some combinations of factor levels appear here. And this approach is usually used due to some logistical or financial constraints. For example, if your incubator isn't large enough to fit all of your samples in a single experiment, or maybe you can't afford to buy all of the reagents needed to do a randomized complete block. A subtype of incomplete block design is a balanced incomplete block design. This concerns how different pairs of the factor levels are assigned to blocks. In a balanced design, each pair of levels appears the same number of times across all blocks, and this allows all of the contrasts to be made between pairs with equal precision, if not directly between individual factor levels. So to consider what the differences between these different types of designs are in practice, let's consider a fictitious experiment where we want to rate the quality of 7 different champagnes using 14 expert taste testers. The constraints of our experiment are that each taster can't rate all 14 champagnes, although that might be ideal from an analysis point of view, makes a randomized complete block design. The rater's ability to rate the champagne declines after they've tried more than 3 champagnes. But they can try up to 3 without any loss of precision in their ability to test. Thus, we define the block size and the maximum number of champagnes that can be assigned to any one taster as 3. So I'd like you to pause now and briefly consider what sort of experiment could you design when you have 7 champagnes and 14 taste testers? Okay, so rating the quality of the champagnes. The first design you might have come up with was to simply assign each of the 7 champagnes to 2 different testers. And you might think there's little need for much else in terms of complexity here, because of course the number of taste testers exceeds the number of champagnes. We can just measure each champagne twice. So the advantage of this approach is it's simple, and our taste testers don't become inebriated because we respect the fewer than 3 champagnes rule. However, with only 1 champagne assigned to each taste tester, we really can't estimate a block effect here, and hence the rating is liable to be biased by, say, individual preferences of each taster. There's also not a whole lot of replication with this design. Each champagne is only rated twice. Given that individual preference in this case might overwhelm the actual signal of quality, here we would really struggle to directly compare any of the champagnes to each other, as each taste tester only drank 1. Or even indirectly compare them by, say, comparing different pairs rated together. Hence, this sort of design is neither an incomplete block design, we can't estimate a block effect here, and it's also not a balanced incomplete block design because it's not an incomplete block design. Now, what if we instead assigned 2 champagnes to each tester? This is still a fairly simple and straightforward design, and we can now estimate a block effect because we have more than 1 champagne rated by each rater. Further, we still maintain our inebriation level in that no tester is assigned more than 3 champagnes, and we have better replication. This sort of design would work fine. You would be able to estimate and do some comparisons here, but it's not the perfect design because this design is unbalanced. If you look on the right of the slide, we have the different pairs of champagnes rated by each rater, and what you can see is that some comparisons happen with greater frequency than others. So for example, I can compare champagne 1 with 3, 4, 5, and 6 because they get rated together by the same rater, but 1 is never paired with 2 and 1 is never paired with 7. So if I wanted to compare champagne 1 and champagne 7 together, I would have to do that by comparing 2 intermediary pairs, say pair 1 and 3 and pair 7 and 3. So our ability to compare champagne 1 and champagne 7 is less precise than our ability to, say, compare champagne 1 to 3, 4, 5, or 6. Hence, this is an incomplete block design, but it's not a balanced incomplete block design. To achieve a balanced incomplete block design, we need to assign actually 3 champagnes to each taster. And this allows each pair of champagnes to appear the same number of times across the whole experiment, even if each pair doesn't appear in each block. This still respects the alcohol tolerance of the tasters, and it also allows for even more replication of each champagne, which is now measured 6 times instead of 4. However, working out how to design such an experiment and assign the champagnes to each taste tester isn't necessarily straightforward. This design is more complicated than the previous two. But luckily, we do have a handy R package called IBD, which includes a function that will produce a design matrix for us. So we don't have to actually sit down on paper and work all of these out as we might have had to in the past. Now the R software can do that for us, which is great. If you want to find more about this software, install it and have a play with it in R. You can also see this link included on the slide for an extended discussion on incomplete block designs. Finally, if you have had much interaction with the field ecology literature, you might have come across a specific type of incomplete block design referred to as a split plot design. Split plot designs came into use because the experiments in ecology are often conducted over physically large space, which makes it either impractical or sometimes even impossible to randomize one of the factors. Consider now an experiment investigating crop irrigation and fertilization together. You can't randomize the effect of irrigation because water can simply seep through the soil. Thus, the plot must be split into two, across which we can then randomize the level of the other factors.